Hi everyone, welcome to the seminar about microscopes, nosema, and tracheal mites, oh my. I'm Bree Price. I'm the Extension Coordinator with the Washington State University Bee Program, and excited to talk today about how to do some microscopy techniques to look at nosema and tracheal mites in your honeybees. More specifically, this seminar will cover the types of microscopes you'll need for each of these diagnostic microscopy techniques, as well as proper handling of microscopes and how to use them. Then we'll talk about the biology of nosema and tracheal mites, looking at what symptoms bees may have, the impact on bee colonies, and possible treatment measures you can use. And most importantly, this seminar will show you how to use a hemocytometer and quantify nosema in your bees, or dissect your bees for tracheal mites and observe any infestation. The types of microscopes you'll use for these microscopy techniques are a stereo microscope, also referred to as a dissection scope, pictured here on the left, or a compound microscope, pictured here on the right. You'll use a stereo microscope for seeing if your bees have tracheal mites, and you'll use a compound microscope for seeing if your bees have nosema. Note that these are both binocular microscopes, meaning they have two eyepieces. There are also monocular microscopes on the market, meaning one eyepiece. It's really just a preference thing, but I personally do prefer binocular microscopes, and I think they're more comfortable to use. Stereo microscopes are what you would use to look at samples that you can hold in your hand, such as a penny, like in this left picture, or a honeybee. A compound microscope is used for samples that are so microscopic you can't see them with your naked eye, such as these mushroom spores pictured here on the right, and you view these samples on a microscope slide. All microscopes have an arm and a base that you'll hold when you're carrying it, and we'll talk more about proper handling in just a few slides. But when you're looking at different parts of a stereo microscope, let's start with the eyepieces. The eyepieces will have a diopter adjustment ring that you can turn to adjust to your vision, as well as this apparatus here the eyepieces are attached to that will widen or narrow to fit the space between your eyes so you can comfortably look through the eyepieces. You'll look through the eyepiece through the objective lens and see your specimen here in your field of view. Your specimen will be sitting on your stage. In this graphic here, there's a stage clip. These help hold whatever your specimen is mounted on, but if your stereo microscope doesn't have a stage clip, that's totally fine. You'll also have a zoom control knob, and you can turn this to zoom in and out and look at your specimen, as well as a focus control knob that will help you sharpen or soften your specimen in your field of view. The typical magnification of a stereo microscope is 10x to 40x. The total magnification is the product of the objective, the eyepiece, and the zoom knob powers. So here's a picture of the objective with a 0.5x power and an eyepiece with a 10x power. You can buy and switch out the objectives or eyepieces for your own purposes if you need stronger or weaker magnification. Stereo microscopes have a fun capability to have a fixed or a continuous zoom. There's usually a switch to change how your zoom knob will rotate. So the fixed function will make it so when you turn your zoom knob, you automatically go to the next dash mark, like the 1 to the 0.8, for example. Or you have a continuous function that will allow you to land somewhere in between the dash marks. This is useful if your field of view of your specimen is just maybe too far away when it's at the lower magnification or lower zoom, and the next one up is just maybe too close. So you can fall somewhere in between and get a nice sweet spot when you're looking at your specimen. When looking at parts of a compound microscope, you'll see that you still have your eyepieces and your diopter adjustment rings, but the objectives and the stage is quite different than a stereo microscope. Your objective lenses, usually there's 4 of them, are attached to a rotational nose piece that you can turn to change the objective. You can see in this graphic that the objectives are different lengths. The longer they are, the stronger magnification they are. Your stage will likely have a stage clip to hold your microscope slide in place. And you have stage controls, which will let you move your stage back and forth or side to side, and you can look at an X and Y field. You also have a coarse and fine focus knob that will let you move your stage up and down to zoom in or out and look at your Z field. And when you're at these higher magnifications or higher objectives, you want to make sure to use the fine focus knob. The coarse focus knob will move your stage in larger increments. So when you're using objectives that are higher in magnification power, they're longer, and you want to make sure to not let them touch your sample. So it's better to use the fine focus knobs when you're using anything more than the 4x objective. The magnification is the product of the objective and the eyepiece powers. Again, the objectives are attached to this rotational nose piece that you can turn to change the objective. Usually the objectives are color-coded. Your eyepiece in this example was 10x, so you would multiply this 10x times your objective. So for example, if we're looking at this red one, this 4x objective, your magnification would be 40x. If you're at the 10x objective, it would be 10 times 10, so you're looking at 100x magnification. The typical magnification for a compound microscope are 40x, 100x, 400x, and 1000x. The 1000x is only used in special cases. For your purposes for looking at nosema, the highest you'll go is 400x. So as I'm talking throughout this seminar on which magnification or which objective you should use, refer back to this slide if you have any confusions. Like I was mentioning earlier, microscopes have an arm and a base that you can hold to carry it around. It's best to support the base with one hand and hold the arm with the other hand so you have a proper hold on the microscope and don't drop it. You want to always carry your microscope upright as well. If your microscope has interchangeable eyepieces, for example, they could fall out. It's best practices to turn off your light after you're done using it. Microscope bulbs can last a very long time if you make sure to turn your light off after you're done using it. If your microscope lenses have dust or debris on them, never use a rough surface like a paper towel or regular bath towel to clean them. Make sure you have a microfiber cloth or microscope lens paper to minimize scratching on the lenses. You can also protect your microscope from dust by covering it. It's best to have it always covered when you're not using it. I've made my own microscope covers with some pieces of fabric. You can also use a bag, or your microscope might come with a cover. And lastly, while you're using your microscope, make sure to not rush your viewing process. Make sure you're using the appropriate focus knobs. Remember those coarse and fine focus knobs with the compound microscope to make sure that you don't scratch your lenses. Adjusting your eyepieces is very important to prevent eye strain or squinting that can lead to headaches or nausea if you're using a microscope for a long period of time. Adjusting your eyepieces to comfortably fit the distance between your eyes is incredibly helpful. Remember, you can turn those diopter adjustment rings to adjust to your vision. Many people can use microscopes with or without prescription glasses. It's really a personal preference, but those diopter adjustment rings will help you adjust to your vision regardless. You can put a small object or ruler under your field of view on your stage and use your focus knobs and your diopter adjustment rings to find a crystal clear image of your sample. Sometimes it's easier to do this if you start with one eye only, like maybe your dominant eye, and then adjust to your non-dominant eye. So here's some steps to break down the process of looking at your sample under a compound microscope, working from the lowest magnification to the highest magnification that you would use for nosema counting. You'll start at the lowest power objective, which is 4x, usually color-coded red. On almost or all compound scopes, the color scheme of the objectives is consistent. You'll place your specimen on your stage, and then using the coarse focus knob, you can make sure to find a crystal clear image of your specimen through your field of view. And when you have that clear image, you can move on to changing your objective lens to the 10x, usually color-coded yellow. Now you'll use the fine focus knob to get that clear image of your sample, and then you can work your way to the 40x objective, which is color-coded blue. This is where you would stop and count your nosema. Again, the last or fourth objective is the 100x objective. Remember that the magnification is the eyepiece times the objective strength. So the 100x objective is 1000x magnification, and you would use a oil called immersion oil to look at your sample. The immersion oil helps reduce any air gaps or light refraction because the objective is so close to the microscope slide. But again, for Nosema, the highest you will use is the 40x objective or 400x magnification. When you're done using your microscope, adjust all the way back to the lowest power and then remove your slide. Now we're going to go through the biology of Nosema species. Nosema is a microsporidia or single-celled fungal parasite. Nosema ceranae and Nosema apis are both species that are known to exist in the United States. Nosema apis has been around for over 100 years, and Nosema ceranae was more recently discovered in 2006. Nosema ceranae is a bit more prevalent and replicates faster than Nosema apis, but they're hard to differentiate morphologically. Unlike Nosema apis, Nosema ceranae is present at similar levels all year round. It's very common to have mixed infections of both of these species at the same time. Another thing to note here is that a recent paper was published reclassifying Nosema to the genus Vairimorpha, but much of beekeeping communities and researchers still refer to Nosema as Nosema, so that's what we'll be using for this seminar. Nosema infection begins when a bee ingests a spore. When a spore is in the ideal conditions, they will explosively replicate. This spore here in the top left shows a tightly wound kind of spring structure in here. And under the ideal conditions, this spring-loaded apparatus here will pierce through a honeybee midgut cell. The cells will then begin to multiply, and when the population becomes high enough, they will burst through the epithelial cells, causing damage to the honeybee gut. All the while, the spores are also taking nutrients from the bees. So the spores are not only damaging cell tissues, they're also taking nutrients from the bees, and the bees are consuming more food, thinking that they're hungry, but they're not becoming satiated. Nosema is often associated with chronic diarrhea or dysentery and inability to absorb carbohydrates and utilize proteins from their food, leading to nutrient deficiencies. These nutrient deficiencies can lead to atrophied hypopharyngeal glands, for example. Young bees might have to skip a brood rearing age since they can't adequately feed developing larvae. They become foragers at a younger age, which means overall they have a shortened lifespan. Queens might tend to lay eggs less, especially if the queen herself becomes infected, causing a reduction in brood production, overall a weakening of the colony. In winter, Nosema levels are often associated with heavy winter losses or inability to overwinter successfully. Overall, Nosema causes a colony's immunity to be compromised. Nosema can be transmitted in a number of ways, such as feeding on contaminated water, performing trophallaxis with an infected bee. If some bees are sick and have dysentery or diarrhea, another bee may ingest Nosema through fecal matter when cleaning the hive. Nosema can also spread colony to colony with robbing or drifting bees. There are no unique field diagnostic symptoms associated with Nosema. If your bees have a Nosema infection, you might see symptoms like declining population, poor honey production, reduced brood production, K-wing. K-wing is when the wings that are normally held together by this little hook called hamuli becomes unhooked. There's a special angle that the bee's wings will sit at. That's referred to as K-wing. You might see an extreme example of dysentery, like here in this picture. But all of these symptoms are not unique to Nosema infection. These symptoms can mean a number of things, such as starvation, viruses, secondary effects from Varroa infestation. So microscopic examination of your bees is really the only accurate way to see if they have Nosema, and we'll cover one of the ways that you can do that microscopy. If you start to notice any symptoms mentioned previously or suspect your bees have nosema, you can send a few samples to a diagnostic lab or quantify the nosema yourself using the skills you learned here today. And if your bee samples show high levels of nosema, there are some treatments that you can use. But really, the best way to manage nosema in your hives is to maintain colonies that have good immunity. Manage your colonies well to reduce stressors, encourage them to be strong and healthy by providing proper nutrition and pest management. But some of the treatments that are available for nosema are fumagillin, which is really the only registered treatment for nosema in the United States. These go by brand names Fumagillin B or Fumadil B. You can also give your colonies a feeding supplement such as Hive Alive or Nosavit. You can also requeen your hive. There is some evidence that queen replacement can help decrease the rates of nosema infection in a colony, comparable to that induced by a fumagillin treatment. However, think about the stress that you're adding to a colony when you're requeening. There's a queenless condition for a short time, there's a lack of brood production for a short time. So you can use whatever treatment you feel is right for your bees. If your colonies have an extreme level of nosema and possibly die from nosema infection, you want to make sure your equipment is disinfected. One way you could do that is if you have any plastic materials such as plastic frames or feeders, you can soak those in a 10% bleach solution for at least 20 minutes. And any bleach solution should gas off after drying in the sun. Another option is to use a blowtorch to scorch the inner hive body or outer hive body and any inner corners of the hive body where contaminated fecal matter might be. If you have any old comb, especially any really old black comb that's been stained with fecal deposits, scrape it all and disinfect the frame itself. Once everything is disinfected, you can put those empty frames that you just scraped back in the hive, and of course the bees will draw out new comb. A good general rule for best practices with beekeeping anyway is to cycle out nearly 30% of your comb every year. So now we're going to start getting more into how to quantify nosema infection. There are many ways that you can quantify nosema using supplies like a mortar and pestle, or for our protocol, we're using things that you'll likely have in your house, such as Ziploc bags and a rolling pin. You'll need a hemocytometer, and in our protocol, we're using disposable hemocytometers, but you can use a reusable one as well. Just make sure that it's clean in between your samples. We also recommend using a clicker counter to keep track of how many bees you're taking out of your sample jar or how many spores you're counting. It's recommended that for nosema diagnostics that you use at least 50 to 100 older bees. These are ones that are likely more infected and can be collected around the hive entrance. If you're also wanting a good representation of everything in your colony, you could do like a 50-50 sample where you collect some bees from your hive entrance and some from your brood frames. Something that's optional but helpful in my opinion is to use scissors or a scalpel and a plate to separate your bee abdomens. Overall, you get less debris in your sample when you separate the abdomens from the body. Remember that nosema is a gut parasite. We have an instructional video on our WSU Bee Program YouTube channel that breaks down the method that we're talking about today, and that video shows a comparison between using whole honeybees and bee abdomens only. Hemocytometers are a specialized tool often used in medical professions or molecular labs to look at blood cells or spores. There are many different types of hemocytometers with different grids or depths in the chambers. The proper type to use for counting nosema is called a Neubauer grid, circled in yellow here. If you buy a reusable hemocytometer or a disposable one that we're using in this presentation, make sure it's that grid type. The disposable ones that we used for a recent diagnostic microscopy workshop are this C-chip brand. When you're looking at your hemocytometer under the microscope, this picture here on the left will be what you're seeing with that lowest magnification with the 4x objective, that 40x magnification. When you zoom in closer, this is what you would see with your 10x objective or 100x magnification. This middle section here has 25 small squares. Of these 25 small squares, you'll count 5 of them. You're going to count the outer 4 corners and the one in the middle. We recommend that you go in a serpentine shape, so you can start at the top left and move to the top right, go down, go to the middle, then the lower left and right. Again, this would be executed by turning those stage control knobs, because you won't actually be moving the actual hemocytometer itself. You'll be moving them with the stage. In each of these squares, you'll count 16 smaller squares. You'll count all the spores that are in these 16 smaller squares. For consistency throughout your counting process, don't count any spores that are in between or outside of the 3 lines on the perimeter. And remember, for counting nosema, you will be using the 40x objective or 400x magnification, so your field of view will be more zoomed in than what you're seeing on this slide. There are different ways to quantify nosema, like I was mentioning, but for our protocol, we're using a Ziploc bag and a rolling pin. Use bees that have been preserved in alcohol. You can transfer 50 to 100 bees to a Ziploc bag. If you were transferring whole bees, you would just transfer those 50 to 100 bees. But if you're separating the abdomens, you would want to use dissection scissors or a scalpel and a plate to collect all of the separated abdomens and then transfer those abdomens to your bag. Close the Ziploc bag tightly, try to squeeze out as much air as possible, and then you can use the rolling pin to crush the contents of the bag until it's flat. Ideally, you want to roll the bag until there's little to no crushing. This step is helping you break apart all that tissue so all the nosema that are in the gut cells are released. Then you would use a graduated cylinder to measure out how many milliliters of water you would need to use, and this depends on how many bees you used. The general rule is 1 milliliter of water per bee or bee abdomen. So if you used 50 bees or bee abdomens, you would use 50 milliliters of water. If you sampled 100, you would use 100 milliliters. When you're reading a graduated cylinder, get down to eye level, and there's a meniscus where the surface tension of the water will collect on the side of the graduated cylinder. At the bottom of that curve is the meniscus. This is what would be the accurate measurement of how much water you have in your graduated cylinder. Pour that water into your bag and shake it around until you create sort of this bee soup or bee slurry, and it'll look something like this. And here I have pictures of a comparison of whole bees or abdomens only. And you can see here that there is a bit of a color difference. If you're using whole bees, you can expect more hair and debris from whatever was on their bodies. Using your compound microscope, which should already be set to that lowest magnification, that 4x objective, I recommend placing an empty hemocytometer on your microscope stage first so you can find your grid lines and make all your manual adjustments to your vision before putting your actual sample on the stage. This will help minimize the sample drying out in that process of getting adjusted to your microscope. Use the coarse focus knob to find your grid lines, and when you found your grid lines, you can adjust the microscope to the 10x objective. Use your fine focus knob to find that upper left quadrant of the hemocytometer within this middle section of 25 squares. And then when you have a clear view of your hemocytometer, this is the step that you could take to gently remove it from your microscope stage and load your sample into your hemocytometer. When you're loading your sample in your hemocytometer, make sure to mix up your bag with your sample in it a little bit more and use a pipette to uptake some of the sample. Avoid any large debris in your pipette and dispense a very small droplet into the hemocytometer chamber. If you're using a reusable hemocytometer, you'll inject your sample into the entrance here on either side. It's kind of like a half circle shape. And If you are using a reusable hemocytometer, you'll just make sure to pipette your sample underneath the cover slip to inject it into the chamber. If you end up dispensing your sample on top of your cover slip or on top of the hemocytometer, you won't be able to see the sample in the grid because the droplet will be on top. It won't be in the chamber. So just be careful about that when you're loading your slide. And then when you're ready, you can place that hemocytometer back on the stage. And again, your microscope will be at the 10x objective at this point. Make sure you refocus your field of view using the fine focus knobs, and then slowly turn the nose piece to the 40x objective or 400x magnification. Again, only use the fine focus knob here because your objective will be incredibly close to your slide, and you want to make sure to not accidentally scratch your objective lens. Use the stage controls to find the upper left quadrant and just move the stage controls as needed, readjusting with your fine focus as you need, and slowly count your Nosema spores. This bottom picture here is what you would see at the 400x objective. So again, at the 10x objective, you have a good view of all 25 squares, but when you're going in to that 40x objective, you're zooming into one of these squares and you'll see 16 smaller squares. Using the clicker counter, you can count every Nosema spore that you see in your squares 1 through 5. If you have a square that has a lot of debris in it, you can use an alternative square right next to it. Scan your eyes in a serpentine shape while counting the spores. And then, like I mentioned, if your spore is partially outside of a cell or somewhere in between those triple lines, just skip it for consistency. So again, you want to serpentine shape view, go, left to right here as you're counting your spores. So once you finish counting all the spores in those 5 squares, you can use this formula to calculate your estimated number of spores per bee. You take the total number of spores you counted times 50,000. For example, the sum of my 5 squares could be 52 spores, and that would mean that I had 2.6 million spores per bee in my sample. Once you're ready to remove your hemocytometer from the stage, make sure to turn your nose piece all the way back to the 4x or even the 10x objective is fine. You just want to create enough room between your slide and your objective lens before you remove your hemocytometer from the stage. Before we move on, let's talk a little bit more about what Nosema looks like. Nosema are very uniformly oval or oblong shaped. They have smooth edges, they have a distinct outline that's a little bit darker. They're sort of the shape of a Tic Tac candy. They are not shaped like a teardrop or lemon shaped. They don't have appendages. And so when you're looking at Nosema in your samples, you're welcome to reference some of these photos to figure out what you're looking at. So going back a little bit more about that formula, once you've counted all your spores and you calculate how many spores per bee exist in your sample, you can figure out what steps you'd like to take in treating your bees or what management decisions you'd like to make. The most accepted economic threshold is 1 million spores per bee. So in my example of 2.6 million spores per bee, that's over that threshold. It would be a good idea for me to try to help my bees, whether that be by feeding them a food supplement like I was mentioning earlier, giving them kind of an extra boost for their immunity, or just making sure that all of their stresses are lower, making sure their Varroa populations are more under control, giving them better nutrition. Overall, I want to increase my colony's health, decrease their stress, so they have a better chance at fighting the infection. So now moving on to tracheal mites. Tracheal mites were introduced into the US in the 1980s. Tracheal mites caused quite a stir for beekeepers as they were first introduced to the US. They were nicknamed the crawling disease because a lot of bees would be found crawling outside the hive. And just a few years later, Varroa came to the US as well. So since then, since the arrival of Varroa, a lot of miticides have been used to suppress Varroa populations, which in turn has sort of created a suppression of tracheal mite populations as well. They're generally considered less of a concern for beekeepers than they were when they were first introduced. Honeybees have an open circulatory system. They have a large tracheal system throughout their body, which spiracles that are open to the environment let oxygen in, and then all of these tracheals ensure that cells within the body receive that oxygen. And tracheal mites take advantage of these spiracles and penetrate through those spiracles and find their way into the bee trachea. So let's now get into tracheal mite biology. Tracheal mites are dependent on honeybee trachea for reproduction and survival. When mature females are ready to move on to a new host, close contact permits the spread of those mites from a bee to another bee. They find suitable hosts by sensing the scent of the cuticular hydrocarbons that young bees have. They're attracted to that smell because they know that that's a suitable host that they can get into those spiracles and move into the trachea through. The tracheal mites spend most of their life inside the honeybee trachea, except for those mature females trying to find a new host. The female mite will lay a handful of eggs. Those eggs hatch and develop into adults. Those males take about 11 to 12 days after hatching to mature. Females take about 14 to 15 days. They'll reproduce. The females will grow into these mature mites and then move on to another host. The female mite needs to locate a new host within 24 hours or it'll die. So like I mentioned, they're very dependent on the honeybee tracheal to survive. Tracheal mite infection often affects the bee's capability to respire or breathe correctly. It reduces their capacity for efficient airflow, wing muscles are often fatigued, and it leads to weakened or sick populations of bees with a reduced lifespan because they can't fly and forage and collect provisions for their hive. Honeybees are thought to be more susceptible to tracheal mites in cooler climates or winter months because of that tendency to cluster together, making an easier transfer of the tracheal mite females. Some of the symptoms that your honeybees might exhibit are a lot of bees crawling around the entrance of the hive. Remember, I was mentioning they were referenced as crawling disease when they were first discovered. Bees are not able to fly because of those fatigued wing muscles. They're also disoriented. They might have a declining population. They might have K-wing. But these symptoms, again, are not unique to tracheal mite infection. A lot of these symptoms could be from poor nutrition, viruses, nosema infection, pesticide exposure. So there are no field diagnostic symptoms that will tell you that your bees definitely have tracheal mites. Microscopic examination is the only accurate way to diagnose them. So here are some pictures of infested or healthy trachea. Normal trachea are clear or colorless or maybe have a very, very pale amber or yellow color to them. Infested trachea will show patchy discoloration or darkening as the mites are feeding and scarring that tracheal tissue. Severely infested trachea have dark brown blotches or black scar tissue on them because of the mites' feeding behavior. Like I was mentioning, close proximity is the way that mites can be passed along to other bees. They can spread colony to colony through drifting. They can spread to new areas through swarms. Beekeepers can inadvertently spread tracheal mites by moving frames of infected bees to new hives or new apiaries. So the best way to manage for tracheal mite infestation, again, just like nosema, is to maintain strong, healthy hives, minimizing stressors. Also, proper control of varroa is helpful. Varroa and tracheal mites are both mites, so many options for varroa may keep tracheal mites at bay as well. If you know you have a tracheal mite problem, these are some more targeted treatment options. You can use things like grease patties. Grease patties are made with vegetable oil and sugar. There's various recipes online that use granulated or powdered sugar, and these patties can be placed at the top of your hive frames. You can also use essential oil as well. Again, there's recipes online. But overall, the grease will disperse onto the bees as they're removing this material from the hive and sort of mask that cuticular hydrocarbon smell that the young bees give off, and the female mites can't locate the bees as easily. Another treatment option is menthol crystals. So these crystals that you can also place at the top of your hive frames usually come in a mesh bag, and the bees will inadvertently breathe these vapors. And as the mites in the trachea are exposed to these vapors, they'll die. And the recommendation for menthol crystals is to leave them in for a couple of weeks as the mites' offspring hatch and develop. You'll be using a stereo microscope to dissect your bees to look for tracheal mites. You'll also need some sort of dish or mounting medium that you can mount your bees on. For our recent microscopy workshop, I made some dishes with hardened beeswax, and that worked just fine. You'll need some insect pins. Essentially, you'll be dissecting the bee to expose the trachea. Therefore, you'll need some tools such as forceps and scissors that will help you do that. It's good if you have some ethanol or isopropyl alcohol is fine as well. You want to keep your dissection area wet with alcohol so your sample doesn't dry up too much. And when you're conducting tracheal mite diagnostics, try to collect younger bees since tracheal mites often prefer those young nurse bees. You can sample right off the brood frames to collect nurse bees. When you're ready to dissect your bee, you'll mount it on its backside on the dish. Use 2 insect pins to mount through the thorax. Try to get as close as you can to the wings or under the wings as possible so you're not damaging the tracheal system. You'll want to use 2 pins, not just one, because if you use just one, your bee might move around as you're manipulating it. Remove the bee abdomen. In this picture here on the middle right, the bee is pinned down just near the wings, the abdomen's removed. This is helpful because if you accidentally squish the abdomen when you're dissecting, your field of view can become littered with your gut contents, and it just makes it harder to see anything. So removing the abdomen is good practice. Then you remove the head and the first pair of legs. This is something you can do with small scissors, a scalpel, or fine forceps, but be careful to not pull too much as your tracheal system might be removed with it. So I would recommend slowly peeling instead of doing like a hard pull. The last step to exposing the honeybee trachea is to remove the collar. You can see part of the trachea when the collar's intact, but you won't expose the whole thing, and there might be mites that are kind of hiding under the area that's under the collar. So this picture here in the middle bottom is this white arrow is pointing to this softer part of the cuticle that holds that collar, and you can lightly pull on that with forceps to remove the collar. Keep your bee nice and saturated with alcohol because if it dries too much, it'll be harder to pull the collar off. And the collar look intact looks like this picture here on the left here, circled with the cursor. So when you're finished with those steps, your dish will look something like this. You have your detached bee head with the front pair of legs, your detached abdomen, your removed collar. It might not come off all in one piece, that's totally fine. And when you're looking at the trachea, they'll be right here at the opening of the thorax. When you're looking for trachea, they're sort of an inverted V shape. This picture here in the middle is a nice outline of the trachea. If your trachea have mites inside, they might have some scarring or some spots. They might be cloudy if they're in an earlier infestation stage, like here in this bottom picture here with the red arrow. But if they are not infested, they don't have any mites inside, they'll be more of a translucent amber color. And this picture here in this top right was done by one of our attendees at our diagnostic microscopy workshop in 2024. She was able to dissect a wonderful specimen and expose both of the trachea, and these are not infested with tracheal mites. So when you're doing diagnostic microscopy for tracheal mites, essentially you're looking for presence or absence. You're not counting like how you would with Nosema. So the general rule is that out of your samples of maybe like 15 to 20 bees or more, if less than 5% of your samples have tracheal mites, then your colony is likely not heavily infested and your bees are okay. If it's more than 5%, you might want to consider a targeted treatment regimen. In this picture here on the left, this is another example of more heavy infestation, scarring. The scarring is a lot darker. The earlier signs of infestation might be more cloudy and white, like on the trachea in the slide before this one. It's not uncommon if one of the bee's trachea is infested and the other isn't. So just keep that in mind as well. It's good to try to look at both trachea when you're doing these diagnostics. Again, like I was mentioning, varroa management and regular use of chemicals for varroa management kind of inadvertently help with keeping tracheal mite populations under control as well. But the symptoms that bees might have when they're infested with tracheal mites aren't unique. These mites might be hidden in plain sight, so this is sort of a fun way to test if your bees have tracheal mites. So as I was mentioning earlier, if you don't have the resources to do your own Nosema diagnostics, there are some diagnostic labs available that you could use. We recommend the Bee Lab in Beltsville, Maryland. You can look them up and find these directions for how to submit your bee samples and what other type of molecular analyses they do. There's also this website here at the bottom of this slide at apiaryinspectors.org with a list of other diagnostic labs in the United States. Here are some other resources for you. So these top two links are some other protocols that look at Nosema using a mortar and pestle. This first one is a PDF showing you the step-by-step, and this video from Oregon State University Extension is a nice step-by-step instructional video. We also made a step-by-step instructional video for how to quantify Nosema using the same method that was explained here in this seminar. If you're interested in watching that, go to our WSU Bee Program YouTube channel or these links here, and you can see also that comparison of using whole bees or abdomens only. Lastly, if you're interested in learning a little bit more about how to dissect your bees for tracheal mites, University of Florida has some great resources. Here is an extension publication and a YouTube video with a step-by-step introduction for how to dissect your bees for tracheal mites. Thank you so much for watching this seminar. If you have any follow-up questions, comments, or concerns, we'd love to hear from you. Please feel free to contact us at bees.wsu.edu/outreach. Thank you.